Hey guys, how's it going? We're going to look at chapter 4 today. And chapter 4 is all about prokaryotes, which is the title, but a little bit more specifically, we're going to look at bacteria. If you recall from chapter 1, I mentioned the two types of prokaryotes are bacteria and archaea, but the archaea are kind of unique and really obscure. So we're really going to focus on the more common and ubiquitous type, which are the bacteria. So in this first section here with the online portion, we're going to focus on some of the basic characteristics, and then we'll talk about how they use those characteristics in our face-to-face section. So we're kind of looking at shapes as a basic aspect of character, grouping patterns, which is what arrangement is, and then looking at structures both on the outside and towards the inside of the cell. Okay, so just real kind of the basic common features of bacteria, which are really one of the most common organisms on Earth. So let's take a look at some of the most common shapes, basic features starting out here. And while there's lots and lots of different species of bacteria, ironically, they can mostly be grouped into some basic shape patterns. There's not a bunch of really elaborate shapes. There's a lot of variation, but that occurs within a couple of basic shape types. So the only confusing part about this is that they use weird terminology to describe what we would otherwise call basic types of shapes. So, for example, a coccus is basically just a circular type shaped cell. Spherical, circular, ball-shaped, all saying the same thing. Coccus is really just a Latin derivative that somehow translates to some circular meaning. Bacillus, similar, is rod-shaped, box-shaped, rectangular, another Latin derivative. So coccus and bacillus are really just fancy words for circular and box-shaped. And then the other third common type are spiral-shaped, and this one matches a little closer. Spirillum means spiral. So circular, rod-shaped, and spiral. Those are the three most common. And we see most bacteria can be kind of loosely placed into one of those. Now there's some other common variations, and these includes more or less intermediates of the three up here. So for example, the first one, Coccobacillus. Rather than being perfectly circular or perfectly rod-shaped, there's an intermediate here. Vibrio, let me just do a quick diagram of this one. Vibrio is sometimes what I call a cucumber shape. It tends to have a fat end and then kind of a skinnier end up here. So it's kind of an intermediate with both cocci and bacillus, but a little bit more oblong. So that's Vibrio. And then finally you have a variation of the spirillum called the spirochete, and I've got a picture of that here. So down here at the bottom we see the spiral spirillum, and then the variation of that is called a spirochete, which is more of a corkscrew type shape. And that's one of the variations we see common in the spirillum. We talked about a parasite that functions with this shape, and that was the Borrelia burgdorferi, the causative agent of Lyme disease. So if you recall that from chapter 1, this particular parasite functions with that corkscrew shape. So anyway, those are the common shapes there. Just go back real quickly, and I've got some pictures coming up here for you, but there's also a figure in the textbook I'll show you. So you'll just want to be familiar with what each shape implies, and basically just be able to pick out some common examples. So let me skip forward and I'll come right back. I've got some shapes here that will go along with the arrangements we're going to talk about next, but you can see individual shapes, and that's all the more I'm going to look to have you identify. I'm not going to, for example, I won't put these two up here and ask you to differentiate between slight variations of the same shape. So just know circular is cocci, bacillus is rod-shaped, Vibrio has a gentle curve to it, spirillum is spiral-shaped, spirochetes are the more twisted corkscrew-like variation of the spirillum, and then branching is another one that I didn't mention. It's not quite as common, but to be fair, that is another one here. So I'm not going to test you over that one, but that is another less common form that we do see. So anyway, as you see from the figure here, there's a lot of variation within each type. Okay, moving on. Another term that you want to be familiar with is a term called pleomorphic, and pleo means many, and morphic loosely translates to shape or character. So really this just means many shapes, and it's a term to describe certain types of bacteria that really don't conform to a particular type of shape. So this is an example here out of the textbook of another parasite we talked about, Helicobacter pylori, causative agent of stomach ulcers. And one of the things about it is that it just never seems to have the same shape twice. and that's just a characteristic of certain cell types, certain species of bacteria, rather. So that's called pleomorphism or pleomorphic bacteria, and you just want to recognize what that means. Now, another way to commonly describe these cells is how they group together. So you can look at the individual cell, or you can look at the pattern of grouping. So we can often then combine the shape terminology with another term for the arrangement. So typically the prefix will describe the grouping or the arrangement, and then the suffix will describe the shape. So for example, looking at the first grouping here. So by the way, just real quick, they can sometimes not have any grouping, in which case they're just referred to as singular and then the shape. So singular cocci would be cocci with no grouping. So that would always just be stated as singular and then shape. So when they start to group then, for example, when we have two cocci grouped together, the prefix diplo is in reference to two, and then cocci then in reference to the shape. So all of these will follow some kind of logic. Again, the terminology is often based on Latin, so it's not common sense, unfortunately. So you have to get familiar with what some of these terms are referring to. So, for example, staphylo cocci. Staphylo loosely translates to grape-like clusters, so these are clustered. Strepto loosely translates to a chain, so you have a chain of cocci. Tetra stands for four, so you have four in this case, kind of like the diplococci, but they're in a doubling pattern there. So tetra means four. There's a lot more that I'm not going to put on here, so just recognize the ones I have here for you, and those are by far the most common. So we see arrangement and then shape going into the same term that describes both simultaneously. Now the bacilli are very similar. You'll have a singular, you'll have a diplo, you'll have a strepto. You don't have a staphylobacilli. That just doesn't occur. So that one's not on here for that reason. And then another unique one in the bacilli that doesn't occur in the cocci are called palisades. And palisades essentially are where they just grow kind of right next to each other. And there is some attachment there, but it's horizontal as opposed to vertical. Or excuse me, I said that backwards. It's vertical as opposed to horizontal. So side to side as opposed to end to end. Okay, so anyway, you can pause this, come back and look at it, and just be familiar with these. Really, these are going to be identification type questions. Matching, for example. Okay, one more note about arrangement is that it's determined by two factors, and you want to recognize that those two factors are the pattern of division, or sometimes referred to as the plane of division, which is essentially where in the cell division occurs. So typically it will occur somewhere in the middle, and this is referred to as the plane of division. Now, when it comes to arrangement, sometimes this plane of division will be consistent in the same orientation with each cell division, and when that happens, we can end up with an arrangement like streptococci, where the pattern of division is the same point in each cell, and as they divide and remain attached, which is the second part we'll talk about, they end up creating a chain like this. Now, sometimes this plane of division will rotate during division, and when that rotates, coupled with attachment still, we can see that the cells grow in a different orientation. So, for example, forming a tetrad. This is one called a sarcine or cuboidal. I didn't have that one on the list there, but this one is shown in this figure where they attach in arrangements of 8 like that. And then staphylococci, what happens here is that the plane of division is random every time. It doesn't ever occur in the same place twice, back to back anyway. And what you end up with is a random association, okay? And that's where you get that grape-like cluster. Now, the other key part is then attachment. So they have to stay attached, obviously, to be in a group. So when it comes to the singular, there's no real attachment mechanism. When it comes to the arrangement grouping, then there's some sort of attachment mechanism there. So we don't need to get into too much more detail than that. Just recognize it has to do with the division plane and the attachment or lack thereof that creates the grouping patterns there. Okay, so you can go ahead and pause it here, take a look at this first review section, and go ahead and continue when you're ready. Okay, next we're going to take a look at the cells in excuse me in more detail. Cells are highly variable, as it says right here. So what we're going to do is kind of fix ourselves on the most common characteristics. Not even going to touch on the extremes. We're going to focus on on the norm and what the average characteristics are. We're going to do this in 3 general stages. We're going to start with the very external features called the appendages. Think of these as like the arms and the legs of microbes. Even though they don't have arms and legs, these are somehow an equivalent. The cell envelope, you can think of it as almost like the skin or the tissue around the outside of the organism. Kind of like our skin holds our body and the fluid inside. You can kind of think of the cell envelope as like that of the bacteria, kind of like the skin. And the internal content is what's inside of the cell, and that would be like our internal body. So our bodies are obviously very different, but that's a way to think about it. Arms and legs, skin, tissue, and then the blood or the fluid inside. So bacterial cells can always be described to some extent with all these features here. So appendages then is a little more of a description for you to write down here, and then the envelope, and then the internal cell contents here. And I put some examples, and we'll talk about each of these as we go forward here. This is figure 4.1 out of your text, and what this tells us, what this shows us, is a illustration of a bacterial cell with lots of the different features we're going to talk about. We'll talk about most all of these here as we go forward, and this is a good reference figure to come back to get a quick description of each of the different layers and each of the different segments that we're going to talk about here. And I'll bounce back to this as we go forward here. So appendages, as I mentioned, are kind of like the arms and legs, but more specifically, they really serve 2 purposes. So starting out with function, they typically will either allow for movement or motility or attachment of some kind, adhesion. So bacteria are often required to move and attach to things, and so these appendages typically serve one of those 2 purposes. And again, there's lots of variations, so we're going to focus on what's most common here. One of the most common appendages is called a flagellum, or flagella for plural. And what this is basically a tail for bacteria. It's a whip-like appendage used as a propulsion device to provide motility. That's your definition there. This is a quick note about how frequently these occur. So as I mentioned, there's a lot of variation, and one of those variations is that some have them and some don't, so they're not always found. And this is a few quick side notes about what types of cell shapes usually have flagella. Now, what I do want to do real quick is have you write down, along with me here, some of the common flagella arrangements. So by arrangement, in this case, we're kind of referring to a similar features in the shapes where in this case, we're talking about how the flagella kind of group along the bacterial cell. So the most common type of arrangement in the flagella is just a single flagella or a lack of arrangement. So in this case, the tail just sticks out one end, kind of like you'd expect it to. And this is what we call a monotrichous. I'll spell that for you here. So we call a mono. Ooh, that's kind of a weird font. How about that? Called a monotrichous flagella. Or you could say flagellum. So one tail. So mono means one. Trichous loosely translates to tail. So that is one of the more common types and really kind of what you'd expect a tail to look like. Now, there's some weird variations of these tails that, something you wouldn't find in other organisms, so really only in these microbes would you see this kind of weird arrangement. But another type is actually called an amphitrichous, I'll spell that, where you have tails at both ends. So amphi, A-M-P-H-I, loosely translates to both. You'll see this in other types of terminology. Amphi is a Latin word that loosely translates to both. And amphitrichous then would be a tail on both ends, okay? And you could say amphitrichous flagella, flagellum, depending on the context. The next is called let me go ahead and spell it first this time. It's called a lopho. Lopho loosely translates to clustering. Lophotrichous then is a cluster of tails. And it would look something like this, where you have a bunch of tails sticking out one end, kind of like that, called lophotrichous. Last one then is called a peritrichous flagella. So peri means around. So peritrichous are flagella that occur around the outside of the cell. So kind of like this. There's no exact number that they will have, but they occur kind of all around. Now you might wonder, how does this even work when you have tails that are all around? They tend to move in one direction, so they'll kind of all synchronize and more effectively push a microbe in one direction. So they don't counteract each other, which it might look like here. They do work in synchrony. So these are the 4 flagella arrangements. Want to recognize those in terms of where the flagella would occur in relation to the name there. Again, it helps if you know the prefixes here: mono, amphi, lopho. You know, I know it's frustrating when we use a dead language like Latin, but you do actually see these show up in other medical terminology, so it's good to know that. And anyway, these are somewhat common. Mono, amphi, and peri are actually fairly common. You won't hear lopho probably as much, but amphi and peri, especially the lesser known. Mono, most people know what that means, but those actually do show up in a lot of terminologies. Okay, next we're going to talk a little bit more about the flagella in terms of how it is attached. So these flagella have 3 components that ultimately allow it to function and then anchor to the membrane of the cell. So we have the basal body, the hook, and the filament. The basal body are a series of rings. It is a series of rings in the membrane that attach it to the cell. The hook then is this angled portion that connects it to the actual tail, which is actually called the filament here, and that would extend all the way out here. So the real tail part of it that we really see is called the filament. Actually, the hook is the connecting part, and the rings anchor. Now, the way the rings and the basal body work is kind of interesting, so I got this quick little snippet from a video, and it shows how these rings are thought to turn. This is an animation, but it's showing the theory behind which they think these work, and that they actually rotate in coordination with each other, much like the gears of a car might do to rotate. That provides torque, which then moves this tail. Now, the tail just kind of moves in a very crude whip-like fashion, but it does provide a source of movement for it here. So that's just a little bit about how it's connected and a quick little visual for how it works there. Interestingly, this is one of the simplest forms of movement in all life. And when I say simple, I put it in air quotes because there's really nothing simple about that. But this is kind of the simplest version of a motility device in all living organisms that you'll find. So kind of an interesting look. And again, it's an animation, but it's kind of a cool take on what they think that would look like. Now, another variation of a flagella, which is not really an arrangement, but more of a unique characterizing feature, is one that I talked about earlier. And there's 2 names for it, so I provided the second one here. Sometimes called a periplasmic flagella, or what we referred to earlier as an axial filament. With this type of flagella, it's actually embedded within the body of a microbe, and this only occurs in the spirochetes. Remember, the spirochetes are the corkscrew bacteria. So in this particular type, you have a unique flagella called a periplasmic or an axial filament that is actually intertwined within the body of the microbe. And what this actually does is it provides a sort of a mechanism of torque, and this is how the microbe twists itself. It's a little bit hard to describe, but this mechanism here, the flagella itself, can actually be contorted or moved, and that actually will drive one end of the microbe in this twisting type motion. So that is the idea behind the periplasmic flagella. The next here is an attachment mechanism. We see that, attachment mechanisms. As I said earlier, the 2 main purposes of an appendage is for attachment or for motility. So the flagella is really the primary motility device, and if a bacteria is going to move in some way outside of natural forces, then it's going to need this particular appendage to do that. When it comes to attachment, however, there are actually at least three mechanisms here, and there's actually a few more that we'll talk about, but these are the 3 true appendages here. And the flagella actually overlaps in both categories. So its primary function is for motility, but in some cases, as a secondary form, it can actually provide attachment. So it is somewhat adhesive and it can stick to things as opposed to being propulsive. But another more common way that bacteria attach are with a mechanism that does this exclusively called the fimbriae. Fimbriae are little fibers that stick out from the cell that allow it to stick to things. And I got a figure of that next. And then the third is called a pilus or a pilus, kind of like potato. It's pronounced both ways. So a pilus or a pilus or a pili for plural. These provide a unique type of attachment for genetic transfer. And we'll talk more about this throughout the semester, but I'll mention it at the end of these examples here. So going back to the fimbriae, as I said, are these bristle-like fibers, and they stick out from the cell, and they attach to things with an adhesive kind of force behind them. So one way I compare these is I compare them to a cockleburl. So I'm pulling up a figure here real quick. Bring this over here for you. Oh, that's cockerel. There we go. You may be familiar with this. They're called cockleburrs, or sometimes called sticktights. And if you walk through fields, especially around here in the Midwest, you probably had these attached to your socks at some point, just like this one here. So I use this as an example because it's similar to the way these fimbriae work in the bacteria. Very different in terms of the types of material that they're made up of, but you can kind of see the analogy there. So they allow for attachment in that kind of way. So these are pretty common. Now, a lot of microbes will use these in different ways. One way is to attach to each other, and then other ways is to attach to a surface. And sometimes it's both. Actually, a lot of times it's actually both. But when they stick to each other, that helps them form groups, and it leads to the formation of what we call biofilms, which is actually different than the arrangement. We'll talk more about biofilms in the face-to-face part, but this type of attachment is necessary for those to form. So they can also attach to a host, and what you're seeing here is a live or a real image, not a live image, but a real image of the intestinal lining, probably of a mouse or rat, with E. coli cells attached to the intestinal lining with those fimbriae. So these are necessary in the natural attachment of microbes to the human body, to animal bodies, natural attachment to the environment out in the soil and in water systems. It's also a mechanism by which pathogens attach to the human body. So pathogens need to cling to a host in order to cause disease, and more often than not, they use fimbriae for that. Now, the third type of attachment mechanism is called a pilus or a pilus, as I mentioned, and this one is a little unique in that it typically provides attachment between two bacterial cells in order to allow for the transfer, or at least the partial transfer, of DNA from one cell to another. So partial transfer of DNA from one cell to another. This transfer of DNA is called conjugation, and it's something that bacteria do uniquely of themselves. The idea is that they will attach to each other, much like two boats anchoring to each other in the ocean so that they can stay close to each other. And then that close attachment, along with some genetic cues from the environment, allow them to transfer DNA back and forth. So there's a lot of factors that go into this, but one of the basic factors is attachment to each other. Imagine two boats in the ocean anchoring next to each other so they can throw supplies back and forth before they drift apart. That's kind of like what this is here. And that process is called conjugation, and we'll talk about that more because it's significant, and it allows for alteration of the DNA between cells, which can cause unique traits to come about in bacteria. And one example of where this is relevant is with antibiotic resistance. This allows for the transfer of resistant genes between bacterial species. Okay, so go ahead and pause it, take a look at part 2, and we'll move on to the next part. Okay, so in addition to appendages, you have surface coatings, which are very similar and sometimes categorized together, although they're just a little different. Let me just go back here real quickly to the figure I mentioned earlier. And where we're at here is we have the appendages shown here in green, the flagella and the fimbriae here. And right now we're looking at surface coatings, which you can see in this kind of off-white creamy color here. And that's kind of surrounding the cell. Next, we're going to talk about the envelope, which are these layers here. But right now we're talking about what's mostly a secreted layer around the cell. It's kind of an oily, waxy layer that bacteria can sometimes secrete. They don't always do this, but when they do, it acts as an extra layer of protection in addition to the cell wall and the cell membrane that we're going to talk about next. So recognize the two major types. One's called an S layer, the other's called a glycocalyx, and that's kind of a weird word, but glyco and calyx or glocalyx are the two words here, or the prefix and suffix. I honestly don't know the calyx origin. I actually haven't looked that one up. Glyco roughly stands for sugars, and that has to do with the fact that it's made of carbohydrate or sugar-like substances. I'm not honestly sure of the origin of this one here. So we look at, again, variation. Some bacteria produce one or the other. In this case, they don't produce both, or some do produce both. Excuse me, I misspoke there. So sometimes it's one or the other, sometimes it's both. And if you go back just real quick again, you'll see in this figure, they're showing a bacteria that actually has both. So this is actually the S layer here, and then this is the surface coating, or the glycocalyx, which would usually be external to that. So the first one, the S layer, is a very thin layer of proteins that is analogous to chainmail or chainmail-like covering. And this is a picture of chainmail down here associated with the knights in their armor. But the idea is that chainmail is thin but strong, and so is the S layer, and it kind of looks like it too. So it's a good analogy. But thin but strong, so it's a good protective layer. One of the interesting things about this, and I have a review question on this, and the review question asks, why was this not discovered early on, or why was this only recently discovered? Let me pause and look at my question here. Okay, the question asks, why have S layers only recently been discovered? And the answer, so it kind of cues that up right here. And the answer is because, as it turns out, in the lab, where most microbes have been traditionally studied under artificial lab conditions, they don't typically produce this. It's only, as it turns out, in the environment, in the more natural conditions, that this particular feature usually shows up. And it has to do with some of the environmental triggers, such as stress, is a very generic kind of way to put it, often in relation to nutrients. But the idea is that we'll talk about this general theme throughout the semester, that micro is an ever-evolving field. In the last 10 to 15 years, it's really been exploding and kind of coming into a whole new era of understanding. So there was kind of what we call the golden age of understanding. They got a lot of the low-hanging fruit, so to speak. They got the easy stuff, relatively speaking. And now, with advanced technology and advanced culture methods, they're figuring out more of the subtleties, which turn out to be fairly significant. Now this one is kind of a random example, but it's a tell as to where we're at. And the fact that this is a very common feature that was only recently discovered is an indication that there's still a lot to learn. And one of the big gaps is understanding them as they are in the lab versus how they are in nature. And the tricky part is that it's difficult to study them naturally, and they're getting better at it. But that's easier said than done. Let's just put it that way. Okay, so that's why that was only recently discovered. And again, the bigger picture is it tells us where we're at in the grand scheme of things, which is still learning quite a bit, and lots of new information is coming out in this particular field all the time. So it's very much an ongoing field of increasing knowledge. So let's take a look at the next part here, and this is the glycocalyxes. And by the way, this is the plural here. Glycocalyx is singular, and they drop the X and add a CES for plural. You gotta love this terminology. So glycocalyxes, plural. So in this case, you have two different types of glycocalyx. And a glycocalyx, by the way, is often made of sugars and certain types of lipids. So that's where the name comes from. Mostly sugar, which is a type of carbohydrate. And this sugary secretion sometimes manifests as a slimy substance, and they call it a slime layer, which is an appropriate name. And then sometimes it manifests as a very thick, very sticky kind of substance, which is called a capsule. So the capsule is thicker and more closely bound to the cell, whereas the slime layer is thinner and much looser and more mucoid in nature. And depending on which type of secretion a bacteria creates, it's referred to as either a slime layer or a capsule, respectively. Um, and again, these are for attachment as well as for protection. So it's another way that microbes can attach. These are sticky. Think of it like putting Play-Doh on the wall or something sticky, and it sticks to the wall with just enough force to kind of keep it there. So it's more of an adhesive mechanism as opposed to a mechanical mechanism with the fimbriae, for example. And this is another side view. This is a figure out of the more recent textbook. This last one here was from a previous textbook. I have a review question that indicates to recognize these two. So I'll use this figure if I'm going to make you identify. Just recognize that this is the looser slime layer and this is the more tightly bound capsule. So that was a quick section. Go ahead and pause it as usual. Take a look at these here and continue when you're ready. Okay, next we're looking at the cell envelope, and I compared this to kind of like the skin of the microbe. So it kind of holds everything internally together. When we look at how the bacteria do this, they do it in kind of three general layers. They have one of the common things found in all living things is called a cell membrane. So this is going to be unique or common rather to all living things. And then with microbes, we'll see a variation of a cell wall, and then in some types we'll see a unique feature called the outer membrane. And these features are going to be combined in a couple different ways to create a couple different unique types of microbes. So the cell envelope is not only part of the structure, but it's also a characterizing feature that differentiate big groups of microbes. So we're not going to talk too much about these in great detail, but I want you to understand the basics. So let me go back here just real quick to that figure I showed you earlier. And I want to show you where we're looking at here in terms of the layering. So the envelope occurs between these three layers here. So this would be the innermost layer, the middle, and then the outermost part of the envelope here. Now these occur external to the surface coatings, which are below the appendages. So again, we've worked from the outside towards the inside here, but we're looking at these three layers here, shown here, here, and here. And I point that out because I'm not necessarily showing them in the exact same order layering. So the cell membrane is actually at the very inner part, and what it does is it acts as the gatekeeper. So you can think of the cell membrane as the gatekeeper for what comes in and out of the cell. And let me pull up another quick figure here for you. We refer to the cell membrane as being selectively permeable. So you can see a common catchphrase here: selectively permeable cell membrane. The reason for this is at the microscopic level, what we see is a pretty tight-knit group of molecules called phospholipids that hold the membrane together, and then we have pores within it called channel proteins that allow certain molecules to pass in and certain molecules to pass out. And then as you see here, some things are rejected from exiting the cell and some things are rejected from entering the cell. So these proteins are selective. Some things come in, some things don't, and this allows for passage. So it's really the proteins that are the gatekeepers. But the membrane itself is said to be selectively permeable. And again, this occurs just above, or at the very base of the envelope, just before you get to the internal contents. Excuse me. Just above that, we have the cell wall. The cell wall is a more rigid structure made of a molecule called peptidoglycan. If we look at peptidoglycan, we see a molecule that to me looks a lot like chain link fence. Chain link fence. Here's a good example of what it looks like, or any of these kind of give you the idea. But it's like chain link fence where it's cross-linked and it has big long horizontal connections followed by occasional vertical cross-links here. Now, really the reason I call it this is because it's strong yet flexible and at the same time porous. So think of a chain link fence. They are pretty strong. You can't just rip them apart. They're not made of tissue paper. But at the same time, they're not solid like a sheet of steel, or a solid piece of metal. They have great big holes in them. So they don't really keep things out very well. Big molecules, yes, kind of like a chain link fence would keep out a big dog theoretically, but they wouldn't keep out real small things like insects and squirrels. So lots of stuff can pass right through it, and therefore it doesn't make it very selectively permeable like the membrane. But what it does do is it provides some strength, some rigidity, and at the same time allows for some flexibility. The cell wall can actually bend and move a little bit, much like a chain link fence would. So that's why I compare it to that. And the molecule here that gives it those characteristics is called peptidoglycan. Peptido is a derivative for peptide, which is in relation to protein. So peptide, protein, and then glycan, similar to glyco, is another reference to sugar. And this is a protein-sugar combination molecule. So it's made up of both types. So peptidoglycan, the only thing I want you to know about this, besides that it's part of the cell wall, is that it's unique only in bacteria. In other words, it's only found in bacteria. That's relevant when it comes to certain types of antibiotics. Penicillin, for example, works on bacteria when it does work because it targets peptidoglycan. And what I mean is that there's a lot of resistance to penicillin, but the way it works when it's able to is that it targets the peptidoglycan cell wall. That's its target in the cell, and that gives it a specificity to only killing bacteria, because that molecule is only found in bacterial cells. So cell membrane, cell wall, and then in some microbes we have a third layer called the outer membrane. This is not found in all microbes. In fact, it's unique to only one type, which we're going to talk about next, and these are called gram-negative bacteria. So the characteristics of the cell envelope, and really to be specific, talking about just the cell wall and just the cell membrane, set up traits that differentiate two major types of microbes. So if you look at the cell wall and the outer membrane, these two features of the envelope set up a difference between two major types of organisms. And these major groups are the gram positives and the gram negatives. Gram is in reference to the guy who figured out the difference between these types of microbes back in the early 1900s, I think it was, late 1800s, I think, somewhere in that neighborhood. Hans Christian Gram was his name. So there's no Latin derivative here. It's just the guy who discovered it. So he named it after himself, Gram, and he determined that you could call one positive and the other negative. It's a very arbitrary naming system. It could have been anything, and he just picked these terms. So gram positive really just is one type of microbe. Gram negative is another type. The names outside of that don't really have much of a translation. But in the gram positives, what you have is a membrane. We see that also in the negatives. So again, that's found in all living things. But with the positives, we see the membrane followed by a very thick cell wall. So they really have only two layers, the membrane and the wall is very thick. So when we look at the gram-positive bacteria, we see two things that stand out. One is a thick cell wall, and in relation to the outer membrane, they don't have one. So the wall is thick and the outer membrane is non-existent. So of the three layers, they have the cell membrane and they have a thick cell wall. Now in the other type, the gram-negatives, we have a different variation. So once again, we have the outer membrane, but here we have a thin cell wall. And the cell wall is comparably thin. So you see here, this is drawn to scale. The cell wall in gram-positives is usually about 10 times thicker, give or take. So much thinner here. So that's one of the characters of a gram-negative, thin cell wall. The other character then is that they have an additional layer called the outer membrane. And this outer membrane is a similar structure to the cell membrane, but it's made up of a slightly different molecule called a lipopolysaccharide. And these lipopolysaccharides, we'll talk about those later because they can actually be toxic, and these can have a negative effect on animals. So really the key thing about the membrane, other than the fact that it's found only in gram-negatives, is that it acts as another layer for selective permeability. So just like the cell membrane is selectively permeable, so is the outer membrane, and this helps filter out certain chemicals from the gram-negatives that the gram-positives can't. So this membrane is actually fairly significant, the outer membrane, in that it really sets these two types apart. In addition, the cell wall over here is much thicker, so that also provides unique features to these gram-positives. So I don't want to go into too much more detail now, but I want you to recognize that these are the two major types of bacteria. And just to clarify, these two are not the only types of microbes, but they make up probably over 95% of all bacteria. So there's actually outlier groups outside of these, but these are by far the more dominant major types of bacteria on Earth, positive and negative types. And these are the differences between them. Okay, this is another figure showing a side view. This is a real microscope image here. It's been artificially colored to show the layering, but you can see these different layers here. So the membrane, the wall, and the outer membrane here. So just a different view of the same thing there. Okay, that's part 4, and we got one more part to go. So last, we're going to look at the internal content of the cell. And when it comes to bacterial cells, prokaryotes, as we mentioned back in chapter 1, there's not much going on. They're pretty simple. So there's not a whole lot to say here other than just to state what is most commonly found, generally speaking, within these cells. So one of the big things, and this is again a characteristic of all cells, so bacteria, eukaryotes, anything, has what's called cytoplasm. And really, cytoplasm is just the fluid portion of a cell, sometimes referred to as a mixing pool for biochemical activities. That's from our book there, that term. The idea is it's fluid. The internal part of a cell is mostly just water, but it's also got a lot of dissolved nutrients, lipids, carbohydrates, proteins, all that stuff dissolved within there, and that's referred to as a mixing pool for metabolism. So another big part then are the ribosomes. We'll talk more about these later. These are somewhat complex, but we can look at them in a real simple way and just in terms of what their function is, and their function is to make protein. So proteins are really important for all living things, and this is where they're made or synthesized, a structure called a ribosome. So for now, just recognize that. And then finally, within a prokaryotic cell like a bacteria, what takes up the most space is the genetic material, the DNA and the RNA. And as you probably know, DNA contains the cellular information, quote unquote. It's kind of like a blueprint for all living things. DNA and RNA are found in all life forms, so this is not unique just to bacteria, but a major portion of a bacterial cell. I made my own little diagram here of a cell. This would be a gram-positive bacterial cell. You can see the membrane and the cell wall here, and it just kind of shows relative proportions of things. These ribosomes, I blew one up to show what it would look like in closer detail. But the DNA usually takes up about that much space in the cell. Ribosomes would be proportional, floating around here in the cell. And then what I showed here is that what happens with RNA is that it's usually processed by ribosomes to make protein. So you might jot this down. I probably won't test you over this now, to be honest with you, but it'll be a topic in the future, so it might help if you connect these here. But essentially, ribosomes, if I could spell it here, ribosomes plus RNA equal protein. And that's a real simple way of putting it. But ribosomes work in conjunction with RNA to make protein. And so that's kind of how those three things connect there. But don't worry about that too much because that'll be a topic later. But that ability to make protein is essential to all living things, and that's kind of why I pointed out, because really these bacterial cells have just the essentials, and these are the minimum things that you need to be a living thing, and that's why we're talking about these here. Okay, last slide here, and then we'll wrap this up. One other feature that's commonly found in bacteria, but not always, are called inclusion bodies. And inclusion bodies are really just storage compartments for excess nutrients. You might think of them as a bacterial fat cell. And when bacteria are naturally in an environment, what happens is the nutrient levels can really fluctuate dramatically. And this is true in the lab conditions as well, pretty much any environment. The problem with bacteria for themselves is that they grow so fast in a lot of cases that they can just suck the nutrients out of their environment, just like overpopulation issues. And they run out of nutrients. So what happens is that some of them are very good at storing away nutrients and then drawing that out over longer periods of time. So it allows them to survive major shifts in the environment. These won't last forever, but they do allow for a longer duration in the environment without nutrients. And that's really all there is to that. So it's a storage that allows for shifts in nutrient availability. By the way, this figure is showing somewhat of an extreme example. This is artificially colored, and these are not usually so massive and numerous in most cells, but in most cells they wouldn't honestly be that visible. So pulled some extreme examples here. This one's out of the textbook to show kind of what these would look like, but they don't usually take up that much space, and they're not pink. That's an artificial coloration. Okay, so that's the end of the first part here. I kept this one to 45 minutes for you. Go ahead and pause, take a look at the last review, and I will see you in a lecture next week. And as always, email me if you have any questions. Have a great day, have a great morning, have a great evening, wherever you're at in life, and I'll see you next week. Goodbye.